Good day. My name is Dr. Essa Suleman, and I'm going to talk to you about novel diagnostic technologies currently being developed at the CSIR for infectious diseases. And this is in the Veterinary Molecular Diagnostics and Vaccines group within the Next Generation Health cluster. So the concept of One Health dictates that human health, animal health, and environmental health are all interconnected. And the aim of and objective of One Health is that it is a collaborative effort between multiple different disciplines to attain optimal health for humans, animals, and the environment. Why is One Health important? One Health is important because every year across the world, there are more than 2.5 billion cases of sickness and 2.7 million deaths caused by zoonotic diseases, of which one of the most recent and well-known is the SARS-CoV-2, which is expected to have its origins in animals and bats. Approximately 60% of human pathogens are zoonotic in nature, meaning that they originate in animals, and there are at least 5 new human diseases every year. And zoonotic diseases also contribute to significant losses in animal production. Globally, there's about 20% reduction in animal production due to the zoonotic diseases. And in order to deal with zoonotic diseases globally, this requires collaborative efforts from multiple different disciplines, whether in veterinary science, human science, and environmental science. But diagnostics is key and critical and central to this concept of One Health, of achieving optimal health for humans, animals, and the environment. There is a current trend in the spectrum of diagnostic testing. Currently, majority of specialized testing is limited to hospitals and advanced laboratories that have state-of-the-art infrastructure, which require highly trained personnel and are expensive to operate. So if you look on the right-hand side, there are hospital-based testing of inpatients in hospital, requires hospital staff. These require expensive equipment, and usually for diseases such as TB, HIV, malaria, looking at HIV viral load, etc. On the left-hand side of the spectrum is point of care or self-testing, and these types of tests can be done by a lay person, and they are rapid. Within 15 to 30 minutes you have a result, and the purpose is for basically self-assessment of disease and then referral to a medical professional. So for example, you have HIV self-testing, pregnancy testing, etc. And the trend as technologies improve is to move from expensive laboratory or hospital-based diagnostic tests to point-of-care based testing that a layperson can perform in the comfort of their own home. Globally, there is a significant demand for development of point-of-care diagnostic technologies based on molecular techniques such as PCR and real-time PCR. And currently, there's about 30% of the global market is point-of-care diagnostics, and molecular diagnostics comprises about 10% of the global market. And both of them grow between 7 to 15% annually. And infectious diseases comprise about 36% of the global demand for molecular diagnostic tests, and particularly point-of-care testing. So there's a significant driver to develop molecular diagnostic tests that are applicable to point-of-care settings. Which brings us to technologies that CSIR is actually developing to enable this. And this is the lab-on-chip molecular diagnostics platform that we are currently developing, which I will spend a little bit of time elaborating on. So current molecular diagnostic technologies are primarily based on either polymerase chain reaction or real-time quantitative PCR, which is, you can see the two technologies on the screen. And this requires samples to be collected from a patient, nucleic acids to be extracted, some sort of amplification and detection, which happens in a laboratory-based environment. And there are significant challenges associated with current molecular diagnostic technologies. So they use techniques such as PCR and real-time PCR. For example, SARS-CoV, the causative agent of COVID-19, the global COVID-19 pandemic, is primarily based on real-time PCR. These techniques are generally laboratory-based, they are expensive, they require highly trained personnel, and they are time-consuming. So it takes hours to days before you get a conclusive result. And the challenge with these kind of technologies is that they are not easily adapted for point-of-care settings. which is where CSIR is developing the lab-on-chip point-of-care molecular diagnostics platform, which aims to integrate 3 critical steps of disease diagnostics: extraction, amplification, and detection. And incorporating these 3 different techniques into a microfluidic device to enable sample prep, and using technologies or novel nucleic acid amplification technologies such as isothermal amplification to enable rapid detection at point of care. And this, combined with a mobile device, results in a device that can be portable and can be taken into point of care, rural settings, etc. Currently, we are focusing on veterinary diseases such as brucella and bovine tuberculosis, but as we expand, the technology can be applied to diseases such as SARS-CoV-2. Isothermal amplification technologies for lab-on-chip applications are extremely useful in that they do not require expensive equipment. A simple water bath or heating block will do. They can be performed at fairly low temperatures ranging from 37 degrees to up to 65 degrees. They are extremely rapid. You have a positive result between 20 minutes to an hour, 20 minutes for RPA and up to an hour for LAMP. And they are highly specific and sensitive. So you have accurate detection of the target pathogen of interest, and they are easily detectable. You can detect a positive reaction via color change, fluorescence, turbidity, etc. And CSIR is looking at loop-mediated isothermal amplification and recombinase polymerase amplification as 2 technologies which for incorporation into the lab-on-chip diagnostics platform. And the ultimate aim of the lab-on-chip platform is that this technology will enable an animal health technologist or a farm owner or field staff to carry out on-site detection of zoonotic and veterinary diseases. So, for example, brucella, bovine TB, African horse sickness, etc. In combination with the lab-on-chip technology, there is a mobile device that facilitates capture of results and reporting of positive cases to the relevant government authorities. So, for example, DAF for veterinary diseases, which is now DLAD. And this can then result in improved vaccination strategies, better disease control, and better surveillance and epidemiology, and also proof of freedom of disease. This will result in increased production and ability to trade. The lab-on-chip diagnostics platform is agnostic, so it can be applied to very different disease profiles. So it can be applied for disease diagnostics and epidemiology, for animal health, human health, and environmental health, and ultimately have an impact on One Health. CSIR is also developing novel diagnostic technologies for SARS-CoV-2, the causative agent of the COVID-19 pandemic. High-resolution MELT strain resolution for epidemiology of SARS-CoV-2 is an important technology that we are developing. There are at least 5 major strains of the novel coronavirus that have been identified and are circulating globally. And it is possible that these strains may differ in virulence, pathogenicity, and may pose additional risks and complications associated with COVID-19. However, to identify these different strains of the virus, is currently the technology used is next generation sequencing technology, which is time consuming. It takes at least 5 to 7 days to sequence a sample and is extremely costly. So it costs at least about 10,000 Rand per sample or per genome in the South African context. And this is limiting because it's expensive and time consuming, so it's difficult to sequence all samples or all positive samples for COVID-19. So alternative technology is required to rapidly identify strains and clade types for SARS-CoV-2 infections. And CSIR is currently developing high-resolution MELT technology to enable rapid diagnostics and strain identification of SARS-CoV-2. CSIR high-resolution MELT assays will identify different strains of SARS-CoV-2 and easily distinguish SARS-CoV-2 from other closely related viruses which a person may possibly be infected with. The HRM technology that we are developing can also be used as a preliminary screening tool to identify positive infections and associated strain types. As mentioned previously, NGS is currently the only technology available for strain resolution, but it is extremely costly and time-consuming, and is not practical in resource-constrained countries such as South Africa and the rest of the African continent. High-resolution MELT is complementary to existing COVID-19 real-time PCR technology and can easily be incorporated into existing testing protocols and regimes. It's faster and cheaper than next-generation sequencing, and HRM will enhance global surveillance and testing of SARS coronavirus 2. And that's it from me. Thank you for your listening. I will now take questions.